Hi, you're listening to Abstract Bioscience, and I'm Michael Pointer. This is a podcast about new and exciting research in biological sciences taking place across the Norwich Research Park and beyond. In this episode, we're going to be looking at new methods being used to monitor crop diseases and increase food security. I haven't had as much homework to do for this one, as this is a project that I've been involved with over the summer, developing and delivering the training workshops that you'll hear about later on. But first, here's some background on why this type of work is so important. And as a note, some of the interviews you'll hear were recorded in noisy greenhouses or windy Ethiopian wheat fields, so there's more background noise than normal. The world relies on wheat. As the leading source of calories in human diets, it supports billions of people across the globe. And as populations grow, we're going to need to increase the amount of wheat that we produce in order to keep up with the increasing demand for food. In the past, attempts to maximise yields have led to the widespread use of very few highly productive varieties, reducing the genetic diversity of wheat and making crops more vulnerable to very large disease outbreaks. Some of the worst diseases affecting the wheat that we grow are caused by rusts. These are fungal pathogens that infect wheat plants around the world. There are 3 types of rust pathogen, each causing its own disease: stem rust, leaf rust, and stripe rust, which is more commonly known as yellow rust because of the bright yellow colour of its spores. Of the 3, yellow rust has the most significant impact, causing losses of roughly $1 billion every year and completely destroying entire crops in the worst hit areas. Most yellow rust fungi reproduce clonally, producing offspring identical to themselves. This makes them relatively slow to change and adapt, and allows wheat to maintain its defences. However, yellow rust populations in Asia commonly reproduce sexually. This shuffles their genomes to create novel strains that can spread rapidly and overwhelm wheat populations that have never seen rusts like them before. One place where we can see the negative impacts of this process is Ethiopia. As the largest wheat producer in sub-Saharan Africa, many millions of people are fed by Ethiopia's wheat. And with 95% of wheat grown on small farms, millions of farmers also rely on it for their livelihoods. The compounding factor is that the prevailing winds arriving in Ethiopia come from Asia, bringing with them the new rust strains that can have a devastating impact on wheat plants that have no defence against them. Wheat rusts are continually changing, like the flu virus or COVID, and they're always moving around. So in any year, the rust disease could be due to a known strain of the pathogen, causing damage but at manageable levels. Or it could be due to a novel strain with the potential to devastate the wheat crop. To prevent epidemics, it's important to monitor which strains are present in the crop, and by doing so, new strains can be detected sooner, and extra resources can be deployed to fight the rust in years when the danger of a large and highly destructive outbreak is high. Dr. Dave Hodson, an expert in rust pathogens and principal scientist at CIMMYT, the International Maize and Wheat Improvement Centre, has spent many years working in Ethiopia.
Rusts are always a major constraint for the wheat farmers. Every season we see issues with wheat rust, both yellow rust but also stem rust. But it's just a continual process of monitoring that's needed here in Ethiopia. Every year you're facing new challenges, new races coming into the country that give problems to farmers. So it's not like you solve the problem in one year. You need a continual monitoring system.
Previously, monitoring work that identified pathogens down to the strain level took a long time. Samples collected in Ethiopia were sent to the US or to Europe to be analysed, and getting the results could take months, during which time disease could take hold. Clearly this was not ideal, and being able to process samples locally and cut the time frame down from months to maybe a couple of days would bring huge benefits. In order to try and make this a reality, in 2017 researchers from the John Innes Centre, in collaboration with CIMMYT, began working on Project Marple. To explain, here's Dr. Diane Saunders, group leader at the John Innes Centre, who leads the Marple project along with Dave Hodson.
Marple stands for Mobile and Real-Time Plant Disease Diagnostics, and it's a way of integrating next-generation sequencing technologies into the genotyping of wheat rust strains so that we can get rapid, real-time information about precisely which strains are present of these wheat rust pathogens in a farmer's field. I think it's really important to know precisely which strain of the pathogen you have in a farmer's field so you can action the most appropriate control. So it's just like when you have a human disease. If you don't know which strain of a pathogen you have, you can't take the most appropriate drug controls. Why we decided to specifically focus on Ethiopia for this Marple Diagnostics technique is because Ethiopia acts as a gateway for wheat rust strains moving into East Africa, particularly yellow rust, where the sexual population originates in Asia and then it moves into East Africa through Ethiopia. And so, of course, they're of high priority for identification of new exotic incursions, which will then move on into neighbouring countries. So it's really important that we have a rapid diagnostics present here in Ethiopia so we can identify quickly those new exotic incursions of yellow rust strains that can move into East Africa.
The Marple Diagnostics Kit is a heavy-duty suitcase that contains all the equipment and materials necessary to perform strain-level identification of wheat rust fungi within 2 days from anywhere without the need for mains power. This is made possible by Marple's use of the MinION portable nanopore sequencing machine, produced by a company called Oxford Nanopore Technologies. For more detail on nanopore sequencing and on the MinION, there's an episode of the Genetics Society podcast, Genetics Unzipped, all about it. But in a nutshell, a nanopore is a tiny hole in a membrane with an electric current passing across it. If a strand of DNA is fed through the hole, the current fluctuates in an interpretable way depending on which base is passing through at that instant. And in this way, the sequence of the whole strand can be read. A MinION contains thousands of nanopores in a machine which fits into the palm of your hand and runs via USB from your computer, removing the need for the very large and very expensive machines needed for other types of DNA sequencing.
The Marple project has been a long-standing project, and it started a number of years ago now when we initially started doing transcriptome sequencing to do high-resolution genotyping of wheat rust pathogens using a collection of samples from all around the world. And this was very advantageous. We got lots of really useful data, but it was based on high-resolution transcriptome sequencing, so we needed to use these large, immovable Illumina sequencing machines. So we wanted to make something that was a bit more portable to open up the chance of doing genomics-based genotyping of rust strains to all around the community. And so we then decided to move our methodology over to the MinION sequencing platform. And so that's how Marple Diagnostics started. And it was really initiated through the want to make this more accessible to everybody, irrespective of access to resources.
After a very successful field trial of the Marple Kit in 2018, the team has established 5 hubs to play host to Marple Kits, as Dave Hodson explained.
So the 5 hubs we're using for the Marple project are really strategically located throughout the main wheat growing areas of Ethiopia. So we're here at Sinana in the barley zone. This is really the core wheat belt of Ethiopia. The other hubs, so we have Kalumsa, where the National Wheat Programme is based. Then we have Holeta, which is where the National Biotechnology Research Centre is based. So there we set them up as like the coordinating centre for the entire Marple project. So Holeta is critical, I think, for coordination and also then capacity building for the other hubs as well into the future. Then at Ambo is the Plant Protection Research Centre. So they're coordinating all the rust surveys throughout Ethiopia. So they have the opportunity to visit many areas, and so again having Marple technology with them is important. And then finally we have Mekelle up in Tigray in the north. So again, that's very strategic because those northern areas we think are also important for as an entry point for new races of wheat rust. So we've seen things that have been in Eritrea and then subsequently moved into Ethiopia. I mean, that's not to say in the future we might be able to expand and add additional hubs, but we see these 5 initial ones as being really critical and very strategic for monitoring wheat rust in Ethiopia.
In recent years, the project has focused on training researchers working in the hubs in the Marple technique, and the most recent round of training workshops was planned for summer 2020, but had to be delayed due to the global coronavirus pandemic. In 2021, international travel was still not possible, but due to the wonders of modern technology, the training was moved online and workshops were conducted remotely from Norwich to the International Livestock Improvement Centre, ILRI, in Addis Ababa.
I think we expect that people should hopefully be able to now adopt the method and run through the whole method from start to end without additional assistance, apart from maybe a little bit of guidance here and there when they might get stuck. But I think we've done a great job in providing them resources that will help them to support them when they want to utilise the method themselves in country.
In the longer term, the team sees Marple building on its past successes and growing to encompass new pathogens and new regions.
We'd like to also, develop the technology so that it's applicable to, stem rust would be the next priority that we'd like to address. So if we can secure funding that would allow us to develop the methodology further, then we would look to expand it to include stem rust. And that would be critically important for Ethiopia. It's a major disease of concern, it's very devastating, and having Marple technology working on stem rust as well would be the ideal solution.
Meanwhile, there's also potential to expand beyond Africa, linking Marple into the early warning forecasting system for rust strains that CIMMET and the Met Office run in South Asia. When reflecting on the entire Marple journey so far, both project leaders picked out innovation and partnership as its major themes.
We have currently 3 key partners who are working together in collaboration to get the Marple Diagnostics method off the ground. So we have the John Innes Centre, where I'm based, and our focus is around the development of the methodology. And then we have CIMMET in Ethiopia, who are working hard to try to help us to implement the methodology in Ethiopia, but also to integrate it into the existing wheat rust early warning system, which is present here in Ethiopia. And then we have EIAR, who are helping us to roll out the method and acting as hosts for a number of the different hubs that we have started to establish here in Ethiopia. I think the relationship between the partners is very easy, so I think everybody gets on extremely well, and we all have our unique role that we can bring to the table. So I think it's really a great partnership that we've established here.
Marple Diagnostics would not be possible without close collaboration between all of its contributing organisations, as well as its several funders from across the globe, and this will continue to be among its strengths as it develops into the future. Thanks to Dr. Diane Saunders and Dr. Dave Hodson, as well as Matt Heaton and Tom Byerly, who recorded parts of those interviews. Videos about the Marple project can be found on the John Innes Centre YouTube channel. I'll also put links to those in the show notes, along with the Marple Diagnostics paper and the Genetics Unzipped episode on nanopore sequencing that I mentioned earlier. This has been Abstract Bioscience, and I'll be back with another episode very soon. In the meantime, show notes, details about the podcast, social media accounts, and links to information about the Norwich Research Park can be found on the Abstract Bioscience website at abstractbioscience.co.uk. The show is written and presented by me, Michael Pointer, and you can contact me at abstract.bioscience.podcast@gmail.com. Our excellent theme music is by the excellent Phoebe Troop. Thanks for listening. Catch you next time.